Hi, my name is Brianna Vargas, and this is my Group Advanced Concepts Presentation 1 for Biochemistry 1. To start off with this presentation, I wanted to add the Part 1 background information that was available on the discussion post. Leucine zipper shown below is an example of a coiled coil. A typical leucine zipper consists of two alpha helixes with leucine residues in about 7 residue intervals. Shown below is one of those intervals. In biochemistry, mutations often are done to study the effects of changing residues in a protein. Electrostatic reactions are where a positively charged side chain interacts with a negatively charged side chain, like the one shown below. Leucine's positive side chain interacting with glutamate's negative side chain. Also for this presentation, I decided to write down all of my responses to the prompts, prompt 1 and 2, and discuss them further in the presentation. For prompt 1, the principles that are involved in holding the two alpha helixes together involve disulfide bonds, ionic bonds, hydrogen bonding, which have hydrophobic interactions, and these interactions allow for strengthened stability of the complex amino acids and enhances electrostatic reactions amongst varying elements. I think it's very important to understand that the helixes have all these different type of bonding mechanisms, and this is why it is diverse and has many components that allow for it to interact with other types of elements. Continuing on with prompt 1's discussion concepts, a possible residue that could replace leucine and maintain the hydrophobic core would be isoleucine, as it is one of the essential amino acids needed in the human body. And it is very similar to leucine in its characteristics and structure. Isoleucine has diverse physiological functions, such as assisting wound healing, detoxification of nitrogenous wastes, stimulating immune functions, and promoting secretion of many hormones. As a soon-to-be medical student and aspiring surgeon, I feel that this is one of the most important amino acids needed in the human body. Not to say that the other ones are not needed, but I do think it's important, especially when there's a lot of infections, and just to promote immune stability, as I do have a thyroid issue, and I feel like this is one of an essential amino acid as it is, but it is very important to sustain, and I would like to do more further research on this amino acid and thyroid stimulating hormones and those interactions and relationships as well. Continuing on with prompt 2 and its discussion concept questions, a possible residue that would destabilize the core would be a hydrophilic residue, as the current residue in discussion is hydrophobic, as we mentioned in the previous slide. A possible residue could be serine. With this presence of serine, the interactions would destabilize the core by breaking down the hydrophobic bonds. Similarly, a different amino acid pair whose side chain could interact in a similar manner would be aspartate and histidine. They are both polar and interact well with one another, as aspartate is more negatively charged and histidine is positively. I thank you for your time and hope that you enjoyed my presentation, and please let me know if you have any questions or any comments, and I look forward to interacting with my fellow classmates and learning more about your discussion posts and how you took this presentation and did it through your own perspectives. Thank you.